Alright guys, in this video we're going to talk about osmosis. So what is osmosis? Osmosis is the movement of water through a semipermeable membrane from a hypotonic to a hypertonic solution until the solutions are isotonic. Alright, let's break this down. So osmosis is the movement of water. That's important to remember. It's our water that's moving, not our solutes, through a semipermeable membrane. So a semipermeable membrane is only going to let certain things through. In this case, water. So only water can get through the membrane. Our solutes cannot get through the membrane. And it's going to be the movement from a hypotonic to a hypertonic solution. A hypotonic solution is going to have a low concentration of solutes. So I remember that there's an O in hypo, low, or no. So we have a low concentration of solutes, or we have no solutes in a hypotonic solution. In a hypertonic solution, we are going to have a high concentration of solutes. And I remember hyper, higher. So hyper, higher. A hypertonic solution is going to have a higher concentration of solutes. And in isotonic solutions, we are going to have an equal concentration. Alright, so let's look at an example. Right here we have a container and it's got water in it. And then we have a semi-permeable membrane. So this is only going to let water through. And if we start adding solutes to the water here, right now we have an equal concentration on each side. So where is our water going to be going? Well, we're going to have an equal movement of water in both directions. Our water is always going to be moving, but right now we have an equal movement in both directions. Now what if we start adding more solutes to this side of the container? This solution is going to be hypotonic. Remember, hypo, low or no. It's going to have a lower concentration than this side. And this side is going to be hypertonic. It has a higher concentration than this side. In osmosis, water moves through a semipermeable membrane from a hypotonic to a hypertonic solution. Alright, so we're going to have a net movement of water towards our hypertonic solution. We have our hypo, our hyper, and this water is going to be moving towards our hypertonic solution. We're still going to have a movement of water in both directions, but we're going to have a greater movement of water, a net movement in the direction of our hypertonic solution. So now our water level is going to change. It's going to go down on this side, up on this side, and now our concentrations are equal again. Now we have isotonic solutions. So how is our water going to be moving now? Well, now we're going to go back to having an equal movement of water in each direction. We have now two isotonic solutions. They have the same concentration. We still have the same number of solutes as we had before on each side, but our water moved, so now we have equal concentrations. All right, now let's look at some examples with red blood cells. So here we have normal red blood cells, and this dark outside is going to be representing our solution, the solution outside. All right, so in a normal red blood cell, if it's placed in an isotonic solution, so what does that mean? Think about that for a second. If it's in an isotonic solution, it's going to have an equal concentration outside and inside. So we're going to have an equal movement of water in both directions. And the shape of our red blood cell is going to stay consistent. But if we have a red blood cell and we place it in a hypertonic solution, remember hyper, higher, so we're going to have a higher concentration of our solutes outside the red blood cells than inside. We are going to have a net movement of water out of the red blood cells. This is going to cause them to crenate. They're going to kind of fold in on themselves. And this happens when we place a red blood cell in a hypertonic solution. the solution is hypertonic to inside the red blood cell. All right, but if we place our red blood cell in a solution that is hypotonic to our solution inside, remember hypo, low or no, so we have a low concentration outside, and then our concentration inside the red blood cells is going to be higher than outside. So we are going to have a net movement of water into the red blood cells. This is going to cause them to expand. They might even explode, and this is called lysing. If a red blood cell is placed in a solution that is hypotonic to what is inside the red blood cell, then it will explode, it will lyse. Another way to think about osmosis is we could say that solutes suck, or water goes where the stuff is. So we have the movement from a lower concentration to a higher concentration. If we have a higher concentration of solutes, well, the solutes suck, so they're going to suck that water in that direction. All right, so osmosis is the movement of water through a semipermeable membrane, so only water can get through, from a hypotonic solution, so a lower concentration or no solutes, to a hypertonic solution, so hyper, higher, so it's going to have a higher concentration of our solutes, until the solutions are isotonic, so they have an equal concentration on each side of the membrane.